Good evening and welcome to the University of Waterloo. My name's Bernie Dunker. I'm the Associate Dean of Research for the Faculty of Science, and we're here tonight to learn more about the Zika virus. To date, Zika has triggered outbreaks in 41 countries, and last month the World Health Organization declared the explosive spread of Zika in the Americas to be a global emergency due to its association with birth defects and the rise in a rare neurological condition causing paralysis and death. Tonight, we brought together a fantastic panel of speakers from the Faculties of Science, Engineering, and Applied Health Sciences to explore the science behind the Zika virus. It's wonderful to see so many of you here tonight. It's not actually the first public lecture that we've had that's been based on a global concern about a viral outbreak. In fact, when the Department of Biology initiated this public lecture series back in 2009, the subject at that time was the H1N1 flu virus. And in 2014, we had another public lecture exploring the science behind the Ebola virus. We're very proud of our relationship with the community and very excited in the interest in current topics dealing with science, health, and technology. So listen closely, think critically, and please ask questions. With that, I'd like to introduce my colleague, Professor Paul Craig from the Faculty of Science, who will be chairing this evening and who will introduce you to our speakers and panel members.
Thank you, Bernie. Like Bernie said, I'm an assistant professor in the biology department here at the University of Waterloo. And first, I'd like to take this opportunity to thank you all for attending tonight. What is sure to be a fascinating and timely look at this recent outbreak of the Zika virus. Tonight, I'm actually playing several different roles. First, I'm here as chair to briefly introduce tonight's topic, and more importantly, to introduce our speakers and panelists. Second, after the presentation, I will be moderating the questions for our panelists. So please, if you could, just hold all your questions until the end and all the presentations are done, and then we'll have an open floor then. And third, I'm a comparative physiologist who studies fish. I don't have a particularly strong background in viruses, so like all of you, I'm really interested in learning a lot more about this current outbreak of Zika. And I'll tell you why. It wasn't long ago, it was only a few weeks ago that I was actually in Florida. And it was for the first time in as many years as I could think of that it was really active spraying going on. And it wasn't just one event, it was multiple days of spraying. I was out on the golf course a couple times, there's a helicopter kept flying by. If you're a golfer, that's irritating. And I asked the golf superintendent, what's going on? And he was like, we're spraying for Zika, or we're spraying for mosquitoes and mosquito control. And at first I was like, great, they're really putting forth a great visible effort to control mosquito populations, which would certainly curb mosquito-borne viruses and diseases. And then I thought, wow, well, I'm just right near, where I was staying was near Fort Myers and Naples. And that's a heavily populated area, and they're spraying constantly, almost every day. And to me, I was like, wow, that's not a really great idea. The scientist in me kind of kicked in. It's like, well, what's these health implications for this? What's the ecological effects of what's going to go on with this? And really, is this constant spraying, is this the right idea to control this kind of disease and this kind of virus? So it's certainly very concerning. And when I came home, I started talking to my friends about my travels, and I was asking them. I had some friends that were going to go down south, and they changed their minds because, well, they heard about the Zika virus. They didn't know a lot about it, but that changed their minds. So there's certainly going to be a lot of economic impacts, I think, with this kind of outbreak in the future, especially with something like the Olympics coming up in 2016 in the summer in Rio. So tonight we'll hopefully address some of these concerns with an examination of what is Zika, how is it transmitted, where it might spread to, where is it spread to now, and how can we combat this virus in not just sort of traditional ways. So with that, I'd like to introduce our two speakers tonight. Since one will follow the other, I'll introduce them both together. Do we have the slide up? I don't know if I can put that. There we go. First, we have Christine DuPont, who is a continuing lecturer here at Waterloo. Christine focuses on genetics, biotechnology, virology, and bacterial pathogenesis, and was a recent recipient of the Excellence in Science Teaching Award in 2013. Following Christine will be Mark O'Coin, who's an associate professor in chemical engineering in the Faculty of Engineering, who has had a direct research experience working with viruses like HIV and is currently investigating the production of complex biologics in cell culture. So please join me in welcoming our speakers tonight, Christine Dupont and Mark O'Coin.
Thank you, Dr. Craig. Thanks for coming out on this spring-like evening. The mosquitoes will be upon us soon. One of the things I know about this audience being from Waterloo is you're probably already very well informed about the Zika virus. And so what we're hoping to do tonight is maybe give you some new information, but also this is going to be about perspective. And the first perspective is that the Zika story is actually much bigger than the Zika virus itself. Zika virus is really just the canary in the coal mine for a lot of emerging diseases that are coming our way. And it's the thing that should have us talking about climate change and some of the impacts of that, our attitude towards genetically modified organisms and vaccines, and as Dr. Craig mentioned, how we're dealing with pest control. So we're going to bring all those things up tonight. My talk is going to revolve around the Zika virus itself, the disease, where the virus came from, and mosquitoes. And so here's Zika virus. And if I pick this virus apart, inside it's got a small genome, and this genome happens to be made up of RNA, so this family of viruses called Flaviviridae, and you can see it includes some really important and nasty viruses, yellow fever virus and dengue virus, as well as our West Nile virus that's here and kind of came along the same route as Zika. They have bypassed DNA. They don't need the DNA. They've gone right to the RNA because that's all you need to make proteins. And so it makes about 10 different proteins, and you'll see 3 of them in the actual structure of the virus. So if I go out a little bit from the genome, there is something called a C protein. It's a single protein, but it forms a shell around the genome. So it's a very pretty geometric shape, kind of looks like a soccer ball. And outside of that protective coat is something called the envelope. And so the envelope consists of, if you can see the blue part, when the virus is leaving the host cell, it actually steals that little membrane from the host cell. So it takes that with it as it's leaving, and just before it's leaving the host cell, it inserts a couple of viral proteins. So one of those is called M for matrix protein, and this one is called the E for envelope protein. And there's a couple important things about this envelope protein. For one, that envelope protein is a major target of the immune response. So if we look at the surface of the virus, not the cut through, we can make antibodies to this envelope virus, and those antibodies will attach to the surface of the virus and prevent that virus from being able to attach to its host cell. So the E protein is an attachment protein. And what this virus attaches to are a variety of cells in the skin. And so one of the signs you're going to see of Zika infection is a rash. Those aren't mosquito bites, that's a rash. Not unique to Zika. You can also find a rash in things like measles and dengue and rubella. But there it is, and that's evidence of infection in the skin. And if we look at the course of the disease itself, well, it starts with the mosquito. She is going to deposit the virus, assuming she's infected, through her saliva. So as she bites, and by the way, it's only the females that bite, and they need to bite because they need some of the nutrients from this rich source of blood to produce eggs. Otherwise, they're out there feeding on plant nectar, just like their male counterparts are. As she bites, she's going to inject some saliva, and along with that comes the virus. And so she's going to do that so she can start pumping blood into her thorax. 2 to 7 days or thereabouts after that bite, most people don't have any symptoms. So 80% of the people who get infected don't have any sort of a symptom. So we're only talking about 20% of people who have And there's a variety of symptoms. Not everybody shows all these, but it ranges from the common ones of headache to the rash. That often includes conjunctivitis. There's no pus associated with it. It's just pink eyes, muscle and joint aches, and fever. But it's short-lived. It lasts an average of about a week, and that's because it takes about a week for the immune system to ramp up and make antibodies that can then bind to that E protein, block the infection, and allow the body to clear the virus. Death is extremely rare, and so in many respects, this is not an important disease. However, there are a couple of complications that can have grave consequences. Guillain-Barré syndrome and the one you've heard of in the news, microcephaly. So I'm going to talk about Guillain-Barré syndrome first. And this is an autoimmune disease. An autoimmune disease happens when the immune system is targeting something that actually belongs to the body. It's not supposed to. The immune system is only supposed to recognize foreign molecules in the body, but for some reason it decides this molecule, in the case of Guillain-Barré, it's the myelin sheath that surrounds neurons. It treats them as if it's something foreign. And so what happens is the myelin sheath, which is kind of like insulation around neurons, degrades. So when the neuron tries to fire an electrical signal, it essentially short circuits. And so that's related to the symptoms that come along with Guillain-Barré, ranging from tingling that usually starts in the extremities and can result in paralysis, rarely death. Most people completely recover from this. Sometimes there's a reactivation. And what we know about Guillain-Barré syndrome, as rare as it is actually not from Zika infection. It's from a host of other infections and even procedures, sometimes surgery, that precipitate this inappropriate immune response. So it's not by any means something that's related just to Zika virus. All right, this is microcephaly, and everyone knows, because you've seen this a lot in the news, it shows up as a reduction in the skull circumference of very young children. And this is, as Dr. Dunker said, was something that triggered the World Health Organization's response to Zika as an international health threat. So a little bit about why that came to be. In Brazil, as it came over into the New World, pre-Zika averages of microcephaly are, and numbers range a lot, but it's less than about 200 per year. And just since October, until March 1st, this comes from the Brazil Ministry of Health, there's been almost 6,000 cases, so that's quite alarming. It turns out when researchers started digging into these cases, that there wasn't a well-defined criteria for microcephaly. So a lot of those are still being investigated, and a lot of dialogue about exactly what the criteria and standardization needs to be for microcephaly. So there's still, the bulk of those are under investigation. But of the ones that have been classified, 641 have been confirmed microcephaly cases that don't appear to be because of physical congenital defects and things like something wrong with the placenta, anything that would prevent a baby's brain from growing properly. So suspect infectious disease. Go in and test to see if Zika is confirmed, and you can see the numbers there. 82 of those 641 is Zika. So that's a rate of about 12.8%. So yeah, that's high, and going to probably get higher as the rest of these cases get evaluated. But that doesn't mean that Zika, of course, is actually causing the microcephaly. So one of the questions, or several of the questions that need to be asked and answered is, can Zika actually get to the fetus? Can it pass through the placenta? Can it get into the amniotic fluid? And can it actually be found in fetal brains where there's microcephaly in place? And the answer to all those things so far in the few cases that have been evaluated is yes. That still doesn't mean that Zika actually causes microcephaly, but the body of evidence is growing. And one of the really important pieces of preliminary information that's come out, this hasn't even been published yet, but it will be, is that human neuroprogenitor cells that you see here, grown in culture, so these are cells that you would normally find in the developing fetal brain, they are virus factories for Zika. So if you infect them with Zika, they are very permissive for production of the virus. And you can see there's the viruses, probably thousands of virus particles in each one of these cells, replicating like crazy. So it can get through and it can act on these cells. And the other interesting aspect of this is these neuronal cells, progenitor cells infected with Zika, actually don't replicate and grow like they should. And that is looking like the mechanism that actually causes the microencephaly, because that's exactly what happens in the brain, is it just fails to grow and develop. So growing body of evidence of Zika being related to microcephaly. But we need, and I put this last, I probably should have put it first, time to evaluate these cases and time to do the research to actually establish that causation. All right, so there's an important story in where the virus came from, because people sometimes think these viruses have come out of nowhere, and that may be because they suddenly hit news headlines and they get sensationalized, but of course there is a story. They did come from somewhere, and it's not like nobody was tracking where they came from. There is a history on where Zika has come from, and it's in the literature. You all know that it's related to mosquitoes. So this is, whoops, not Angelina Jolie. This is a female Aedes aegypti, and that's an important mosquito that you're going to see, emerging from her underwater pupa, and that's an actual photograph. It's a beautiful photograph, but what it reminded me of as soon as I saw it was Angelina Jolie playing Grendel's mother in Beowulf, right? Just beautiful, but really quite menacing. All right, so here's the story. 1947, there's a research station in Uganda, and it's still there. It's in the Zika forest of Uganda. That's where the virus gets its name from. And what they do at this research station is they put up monkeys that don't belong to Africa and put them up there in Africa and see what kind of viruses that they pick up. And that's how they found Zika. All right, so Zika is a flavivirus, and we know there's some really important viruses in this family, and that they're often transmitted by mosquitoes. So they started looking for this virus in the native forest mosquitoes. And indeed, they did find it really quickly, about a year later. So, red flag number one. This is a flavivirus. It's transmitted by mosquitoes. There's a mosquito-monkey cycle going on. Where have we seen this before? Dengue, yellow fever. Can it infect humans, this one, though? And yes, it can. There was a single confirmed case of a person who actually was working in the research station who was infected with Zika, classic Zika syndrome. And during that time, they were also looking to see where else might humans have been exposed to this virus. And so they were looking for antibody to the virus. And they found it widespread in parts of Africa and Southeast Asia. People had been exposed to this virus probably for a very long time. That's not so surprising because there's monkeys in these places and there's certainly mosquitoes in these places, all right? But it's widespread. Another red flag, 1966, Aedes aegypti. This mosquito is not a native forest mosquito. Another red flag. This virus can move from mosquito to mosquito to mosquito, and this mosquito is widely distributed. We did that, right? So another red flag. And the virus did move. A couple of outbreaks, 2007 in Micronesia. This is an area that has never seen Zika virus before, and there's no monkeys here either. So it's infecting humans and mosquitoes, human to human transmission, and we saw that again as it started to spread to that area of the Pacific. Again, no monkeys, and from there, Brazil. Okay. Well, Brazil has monkeys, but that's not who brought it there. All right, so the mosquitoes are important in this scenario, and it's really an interesting part of the scenario. The virus's ecology is intrinsically linked to the mosquito ecology, and some of these mosquitoes you're going to see are really linked to what we do. So meet Aedes aegypti, whoops, and its cousin Aedes albopictus, and fancy mosquitoes compared to our one of our native species. This is Culex pipiens, and they all carry some important viruses. Chikungunya virus, yeah, you can think about saying that as I'm talking. It doesn't belong to Flaviviridae, it's a different group, but it's an important virus as well that's carried by these mosquitoes. Aedes aegypti, originally native to Africa, but it's spread in wide parts of the world. It's an invasive species, okay? And it's an impressive mosquito. Make sure you ask Dr. James about this mosquito. This one is also widespread in the world, but it's actually only native to Southeast Asia. So it's an invasive species in lots of other parts of the world. So as pretty as these mosquitoes are, and as impressive as they are, we probably really should be killing them. The reason that I put Culex pipiens up here, one of our native mosquitoes, is because the way that we, I think about mosquitoes and the way we deal with mosquitoes and try to protect ourselves from mosquitoes here in southern Ontario is very different to what you have to do when you meet one of the Aedes mosquitoes. So I'm going to go through a few things rather quickly. Our mosquitoes, everybody knows they come out at dusk and dawn, and so you either go in your house and protect yourself with your screens and your doors, or if you're going to be outside, cover up, get your DEET on, but look at the Aedes mosquitoes, daytime biters. So we have to change the way we behave if we're going to be in areas where these mosquitoes are, or if those mosquitoes come to us. Our native Culex mosquito likes to feed on birds. That's its preferred blood source, and so you'll find it intimately associated with forests, because that's where the birds are now. If you get into the pathway there, it will happily bite you as well, but you are not its preferred meal. Aedes albopictus feeds on a wide variety of vertebrates, and it transits between the forest and urban areas. And this is where Aedes gets quite impressive. It loves humans. It prefers humans over other sources of blood meals, and it's a multiple biter. So I said that most female mosquitoes are only going to bite when they're about to develop eggs because they need those nutrients, and this one, it looks like, does between meal snacks on blood. So it can infect a lot of people because it can bite quick, lots of people, and not just for the purposes of laying eggs. And it's an urbanized mosquito. It has essentially domesticated itself, habituated itself to us in our urban environments. And some of what that urban environment looks like in places of the world where this mosquito has moved into is lots of containers, lots of plastic. Collection of water is often done in a variety of different ways, and mosquito eggs are easy to find in these places, including on plants and in pots and things. Interesting thing about Aedes aegypti mosquitoes is, as opposed to our native mosquitoes that lay eggs on the surface of the water, they have sticky black eggs that they lay just around the surface intersection of where the water is going to be. And so all kinds of containers, including in our disposable society where there's places that don't have garbage system pickup, you'll find lots of containers. And we're always told that, dump the water out of sitting containers, and that will flush out our native mosquito larvae and eggs, but Aedes aegypti eggs will still be in the container. And if you look carefully, you might be able to see some of the rims of the eggs around here. So you can dump the water out, but if you leave the garbage there, the next rain is just going to fill it out, and you're going to have a brood of new mosquitoes hatching from that. So it's a different kind of mosquito, and we have to deal with it differently. The eggs can also withstand desiccation, so we tend to think of mosquitoes being present in areas that are thickly forested or swampy areas and things like this, but this is a very arid area. This is a shanty town in Mexico, so just imagine what's going to happen when the El Nino rains come and fill up all these tires and containers. So the story really is that the virus didn't come to us. In many ways, we have come to the virus. So where it actually came from and how that transition kind of happens, and you see the same thing with yellow virus and dengue and something similar with West Nile virus, just take the monkeys out and put birds in its place. there's in the forest, in the jungle, native jungle mosquitoes and monkeys. And as long as both of those species can replicate the virus to a high enough level, a titer, then they can transmit back and forth both ways, right? So the cycle continues. How do people get in here? Well, people enter into this cycle one way or another when they start to work, invade, deforest areas. Going into the forest for hunting, for example, for the large bushmeat industry that exists in many places around the world, and really, they're inadvertent victims of some of these diseases. And there's a couple of scenarios that can happen after this. If humans can't replicate that virus to a particular threshold level, then you actually can't pass it back to any of the naive jungle mosquitoes. So we're still in the natural setting, except humans are starting to move in to this natural habitat and ecology of the mosquito and the monkey. If we don't replicate this to a high enough titer, well, we could still get sick. This is what happens with West Nile virus, just put a bird there instead. But humans become a dead-end host, okay? They can't continue a cycle amongst humans. And that's known as a spillover event, by the way. When an infectious agent that's in a natural animal reservoir moves into some other species where you wouldn't normally find it, in this case, that's the humans. So the other scenario is that if people can replicate this to a high enough titer, we can transmit it back to mosquitoes, naive, uninfected mosquitoes, and in areas where you've got people interfering, living closely, deforestation for crop purposes, for example, in these areas where there's a monkey-mosquito cycle, you can actually get both going. And if you've got enough people that are transmitting it to the mosquitoes in this cycle, then you really don't need the monkeys there to keep it going. Remember we said no monkeys? And so this is what's happened in Brazil and some of the other outbreaks. What's made this happen to become urban, though, is the movement away from a native forest species into this, urban species. And I'm picking on Aedes aegypti, but Albopictus has the same kind of scenario. So now we've got a scenario of people and mosquitoes, and you don't need the monkeys, and that's what we're seeing in Brazil, causing an epidemic. So areas where you've got endemic cycles going, shown in purple, and the virus got on a boat through an infected mosquito or probably through an infected person, and we'll never know who that is or which one happened, but brought it to Brazil, and it spread like wildfire epidemic. All right, so what's an epidemic? An epidemic is related to immune status. Really, it's when an infectious agent enters a population with no previous immunity, and it spreads. And Zika can spread really quickly. Epidemics don't imply that there's a particular level of death or morbidity and sickness within a population. It just means that it's spread and you can detect it, and there is people getting sick, but it doesn't mean they're getting really, really sick. And that's the case with Zika. Most people, it's a very mild infection. All right, so the scenario or the cartoon, if I was to draw it out, kind of looks like this, where if you've got a population with no immune memory, never seen the virus before, there's no protection, it's going to spread, lots of people are going to get infected. So if you think about what's happening right now in the Americas, lots of people are getting exposed. And that means that the virus is probably going to hang around for a long time. It's in the mosquitoes, it's in the people, it's going to become endemic. But these people have been exposed. They've got immune memory now, and they will be protected from subsequent infection. And really what this is saying is that the best protection from Zika is probably getting Zika. So go get it now. Go to Florida. All right. Aedes aegypti and Albopictus. So, what are we in for? This is the predicted distribution of these mosquitoes based on a preferred habitat. And actually, these mosquitoes have kind of filled this up pretty well. And if you can see it, if I'm not in the way of it, here we are down here. And we have actually found some Albopictus in Ontario. It's not established, but it is probably going to if things continue the same way with climate change. And what I really like about these maps is you can overlay them with areas of not only where these mosquitoes are, but where the diseases are, like Zika and yellow fever, chikungunya, and you can come up with a retirement plan. And with that, I'm going to retire, and I'll turn it over to Dr. O'Coin.
We'll start off a little bit about talking about areas around the Zika virus, okay? Themes that are not necessarily directly related, but really are. And so the first one I'm going to talk about is globalization, global warming, and changing ecosystems. And to begin with is, I'd like to ask, do you think that Chris is going to be safe with her retirement plan, right? Has she chosen appropriately? Are the maps really indicative of what's going to happen when she decides to retire? You see, a year ago, the CDC presented this map as the distribution of Zika virus in the world. And you can see that it's really located primarily in Brazil, some parts of Africa, India, and Indonesia. Well, a year later, and with following it a lot more, we can see that these are all reported cases of where the Zika virus is. Now, we've talked about how the Zika virus is spread via Aedes aegypti and Aedes albopictus. We can also say that it's not likely that these mosquitoes bit these people in Canada, because this is not a location where those mosquitoes will survive. Well, so how did these people get infected? Well, because us North Americans, we really like our tropical holidays. We like to go into the tropical regions, the Caribbeans, hotter regions, in order to relax and relieve all of our stresses. However, maybe it's more stressful to travel down there now that there's the talk about Zika. Fact is, there's a lot of talk about Zika, and everybody's worried, and it's probably mostly because of the cases of microcephaly. Okay, it's actually a very scary visual problem, to the point where airlines are now allowing you to refund your tickets or change your flight plans if you're a woman and you have a partner, and you don't want to travel to those regions in case that you were at a point in time where you thought you might be getting pregnant. The question is this overreacting? Because really, on Canada's watch list, there's a number of tropical diseases and mosquito-borne diseases that are very serious as well, right? Zika, chikungunya, malaria, Rift Valley fever, Venezuelan equine encephalitis, West Nile, dengue, yellow fever, Japanese encephalitis. A lot of these things are pretty nasty, right? And really, if you look at that category, a number of them are actually mosquito-borne with Aedes aegypti, or Aedes species, okay? So the question is, still, are they overreacting? Well, possibly. Because of all those mosquito-borne illnesses, only one is now thought to be transmitted sexually, and that's the Zika virus. And this actually was published in 2011 for the first time, that this was a possibility, and they found this out after researchers came back from studying mosquitoes in 2008, and they were spread to their partner once they came back. And now, the World Health Organization is also confirming that sexual transmission of the Zika virus is much more common than thought. So, it's likely, or it's quite possible, that spread beyond the tropical regions will happen. But just because it's sexually transmitted doesn't mean that it's going to necessarily move all over the place. Okay? Hopefully, maybe, we as humans, we might not be as, well, should we say adventurous or seeking out as many partners as our friend Aedes aegypti. Okay? And maybe if we protect ourselves through contraception, we can actually reduce the person-to-person transmission. So that leaves us with the question of whether or not we're safe from the mosquitoes. So it's always back to those mosquitoes. Is global warming helping us in this case? Probably not. Okay? We're reaching some of the highest temperatures we've seen in a number of years. And that means that we could be expanding the habitat for these mosquitoes. In fact, there's a study that was done recently that shows, or that's estimating how Culex pipiens, our mosquito in Canada, will actually move more northerly over the next 100 years. You might say, well, Culex pipiens is not Aedes aegypti, it's not Aedes albopictus, but I'll forward you this one. Okay? With global warming, you're actually going to start spreading the range of these mosquitoes. So one, you're going to have more mosquito overlap. Okay? And when you have more mosquito overlap, you actually have chances of switching mosquito species from which the virus is actually in. And two, we got to be careful because we now have some studies that point that Culex pipiens can also transmit the Zika virus. It can also be a host. So from Aedes aegypti to Culex pipiens to us. Okay. So that brings us to the idea of, well, maybe we need to do some pest control. Well, this is quite interesting because with pest control, we're going to try to eliminate mosquitoes and we're going to try to eliminate the virus. Well, when Chris was talking before, what did she say about the virus becoming endemic and possibly that being the best case scenario? Because once it's endemic, we might actually gain immunity against it. So let us actually get infected with the Zika virus, and those who survive will be that much stronger. Okay, well, that might be the way to go. Because what happens when we actually try to eliminate it? Our mosquitoes and our virus, and for some reason or another, one of them actually remains alive. Well, there's the potential for a future outbreak. Well, what happens to the population when they don't see this endemic virus? Well, they don't gain that immunity. Okay? And so because you don't have that immunity, you might actually have a greater risk against those viral outbreaks and the disease. And so when we talk about pest control, really, if we're going to talk about pest control, if we're going to talk about eliminating these mosquitoes, we really also have to talk about vaccination. Because both of them together can actually lead to better protection. Okay, but first on to pest control. For mosquito control, from what I understand, there's two real main schools of thought. One is go after the adults or go after the larvae. Okay? Kill off the adults, they won't bite, you won't have the spread of disease. Kill off the larvae before they even become adults, they'll never bite in the first place. So you really have these two stages. One, that larval stage, which is really in those water sources, those water reservoirs, all those beautiful pictures that Chris showed about where mosquitoes will breed. And once they're out and about and flying around. You can try to destroy all the larvae, and the pupa. You can put insecticides in the water. You can put, these insecticides can be chemical insecticides, it can be biological insecticides. But will you be able to find all of the water reservoirs around? The answer is no. In fact, Aedes aegypti can breed in pretty much any little container that has water. Bottle caps, for example. Right? How are you going to find every single bottle cap? There's a lot of bottle caps out there. How many people drink water from a bottle? Oh, that's recyclable. Another major initiative against mosquitoes is bed netting, right? And for malaria, this is probably one of the biggest initiatives. And this is great if you're trying to protect against mosquitoes that are going to bite at night. But Aedes aegypti, our major perpetrator of our Zika virus, well, they're a day biter. And I don't think many of us are going to go walking around in bed netting during the day. Our moderator, our chair, told us about his golfing adventures and how he saw fogging, right? Ultra low volume spraying. What he said was it's a very visible thing that they were doing to try and attack those mosquitoes. It is very visible, and people feel reassured that something is being done when outbreaks are happening. But the fact is, it's probably one of the least effective methods in controlling mosquito populations. In fact, not only is it not very effective, okay, it's not necessarily specific to the mosquitoes. It can also target the mosquito predators, okay? It's natural predators, those that are actually keeping the mosquito populations at bay. It's also breeding resistance to mosquitoes. And it's also probably one of the most costly ways of doing it. And because of that, it's not sustainable. And if it's not sustainable, we'll do it for a short period of time. Hopefully there's going to be an impact in that small duration, but long term, they'll come back. So what more can be done? What else can be done? And something that could be done that's more specific. Well, here I'd like to introduce another player. I don't think we've talked about enough different organisms so far. Well, let's introduce another one. Let's introduce you to Wolbachia. Wolbachia is pretty cool because here we're looking at infiltrating the mosquito population. Wolbachia was first discovered in 1924 in Culex pipiens. Okay? And what it is a bacteria that actually needs to live within insect cells. It's almost like a symbiotic organism. It's not going to kill off the insect, but it will live within its cells. It's almost a little bit parasitic, but it's not really to the detriment of the insect that it's occupying. Let me tell you a few things about Wolbachia. There's researchers that have looked into trying to get Wolbachia into Aedes aegypti. It wasn't an easy thing for them to do, but after a number of trials, they were able to do it. They introduced it through the egg, to the mosquito eggs, and eventually got Aedes aegypti that had Wolbachia in them. Now, male carrying Wolbachia mosquitoes, they can mate with wild-type males or males that also have Wolbachia. And their offspring will all have Wolbachia in them. Male mosquitoes carrying Wolbachia will not be able to mate with successfully for producing offspring with wild-type mosquitoes, female mosquitoes. The other thing that you should know is that mosquitoes carrying Wolbachia can't reproduce or replicate certain viruses very well, including dengue, which is closely related to Zika. What this means is that now you have a female mosquito that contains dengue, or no, that contains Wolbachia, that will not actually lead to the spread of the disease because it can't spread the virus, because the virus will not replicate within it. So this leads to two different strategies to actually combat mosquitoes. The first one is to release a whole bunch of Wolbachia-carrying males and let them mate with wild-type mosquitoes. When they do that, the offspring will just die off. Okay? Now, the problem with this strategy, or may or may not be a problem, is that you have to continuously supply those male mosquitoes. Okay? The other way is to allow Wolbachia-carrying females out into the population. So you release a whole bunch of Wolbachia-carrying female mosquitoes. They will mate with wild-type males, carrying Wolbachia or not. And the result is that their offspring will not be able to replicate the virus. And so, yes, you'll still have a lot of mosquitoes out there, and yes, they'll still bite you, but they won't spread the virus. Now, this is an example of maybe an engineered system, but not a genetically engineered system. Because these organisms naturally infect these cells. All the scientists did in this case was put them together. Put them in the same room and let them dance. But there are some other options very similar, and arguably not all that different, but based on genetically modified organisms. So genetically modified organisms, what they're going to do is oh my goodness, my voice. Sorry. I feel like I'm 15 again. Genetically modified organisms, you're actually going to manipulate the genetics of the mosquito. You're going to introduce a new gene. One example of this is what's known as the release of insects with a dominant lethal gene, also known as riddle. In a first case here, you have a dominant lethal carrying male mosquito, which will mate with a wild-type female. In that dominant lethal carrying male, he'll carry a gene that's been inserted that will render the offspring female flightless. As a result, that female mosquito will not be successful in life later on. Another example, again, with the dominant lethal carrying male. That dominant carrying lethal male has another type of gene that will actually only turn on once in the larval stage. And what it will do is that gene will turn on in the offspring and lead ultimately to the mosquito death. Both of these will require the release of male modified mosquitoes. But these male modified mosquitoes will eventually die, and these male mosquitoes are also ones that don't bite. Another strategy is using the homing endonuclease genes. So again, you're genetically modifying your mosquito. So you're actually introducing a new gene, a gene that was not necessarily present. This gene, though, is a very special gene. It encodes for an enzyme. An enzyme is one of these really cool functional proteins that does something neat. What does it do? It actually cuts DNA. And when it cuts that DNA inside the cell is going to try and repair that DNA, because the cells don't like having broken DNA inside of them. And when they repair it, they actually repair it with another copy of the same gene. What does this mean? It means that we can ensure that whatever gene is being inserted will be actually expressed in the progeny. And so what they've used this for is to actually skew the sex-gene ratio of the offspring, such that no female offsprings will be produced. So only male offsprings will be produced. And this is a self-perpetuating system, so you don't actually have to release a whole bunch of males, because you're producing new males that are going to be able to do the exact same job. And eventually you're going to eliminate all of the females. And if you eliminate all the females, well, then you're pretty much safer than you were yesterday. So I come to my last bit, vaccination. Well, not for all flaviviruses. I don't know if you remember at the beginning of the talk, we talked about the family Flaviviridae. This family of viruses contain viruses like dengue, like yellow fever, Zika, West Nile. And the fact is, we don't have vaccines for all of these. In fact, for Zika, there's none. For dengue, there's none. Well, for dengue, I should never have written that. There's none that are fully approved in Canada, so we have to wait. There is one that's approved, and that one is approved in Mexico. It's a really recent approval. And it's not based on dengue, it's actually based on the yellow fever vaccine. So I guess I should tell you a little bit about what the yellow fever vaccine is. The yellow fever vaccine is a live attenuated virus vaccine. So what do? They inject people with yellow fever virus, and they gain an immunity to the virus based on the full replicative virus. The attenuated part is that in order to say that this is the right thing to do, they want to make sure that you didn't get the same type of sickness, the same type of illness that you would get with natural yellow fever virus. And so what they did was they took it from a person. In fact, it was isolated in 1927 from a Ghanaian, and it was transmitted to a monkey. From the monkey, it was transferred to mice, not before killing a number of people, actually. It wasn't just a, well, let's just take it and put it there. It was like, come here, oh, one guy died, oh, another guy died, another guy died. Until yellow fever is nasty, right? Until Max Steeler came around, he transferred it to mice, and then after that, he passaged it in chick embryo tissue, 76 passages. So they actually, what they do is they grow it to a certain amount, and then they put it into more cells, and they make more of it, and then they put it into new cells and pass more of it, make more. A lot of times, right? So they went 76 passages in chick embryo tissue. It still wasn't attenuated. And then they had to passage it 100 times in tissue without, with the neurological tissue removed. And when they passaged it over 100 times in the neurological, in the tissues with the neurological tissue removed, then the neurovirulence was reduced. The neurovirulence is what they didn't want to have. That's the bad part of the disease. After all those passages, they got an attenuated strain. And that's the basis of the yellow fever vaccine. For dengue virus, the most clinically advanced dengue virus is based on the attenuated yellow fever vaccine. And what they did is they made a chimeric virus, Remember, these viruses are very, very similar to each other. And so what they did was they removed the 17D structural protein. 17D is the attenuated strain of the yellow fever virus. So they replaced its structural proteins with that of equivalent regions in the dengue virus, and that's what they're using as the dengue vaccine for all 4 serotypes. So the question is, because dengue, yellow fever, and Zika are so closely related, could we have a chimeric Zika vaccine? Well, in fact, it's a possibility. In fact, Sanofi has said that they're going to use that strategy to try and build that vaccine. Do we have the vaccine today? Are we going to get the vaccine tomorrow? Don't count on it. But are we in a good position to try and find a vaccine for it? I believe so. Then the question is, well, just because we have a vaccine doesn't mean people will actually use it. Would you use it? I always ask somebody when we talk about vaccines, a vaccine covers many different types of compounds. If you talk about the flu vaccine, we're not talking about a live virus when we talk about the flu vaccine. I would say that the flu vaccine, from a certain standpoint, is much safer than a live attenuated vaccine, right? Because a live attenuated vaccine, there's always a chance that that virus, okay, becomes more virulent and cause sickness. So it's not a given that people will actually use the vaccine if it's created. But it's good to know that there might be one available pretty soon. So now we've come full circle, and we are at your disposal. We will be coming up here. I'll invite the panelists and Christine. But that's probably your job, right? Okay. And you can pepper us with questions, poke us like a bunch of mosquitoes.
I'd first like to introduce Craig Janes, who's a professor in the Faculty of Health Sciences, whose research focuses on infectious disease and global health governance. Craig is also a recipient of the National Medal of Honor by the government of Mongolia for his contributions to developing the health sector in Mongolia. Our other panelist is, Kim Cuddington, an associate professor in the Department of Biology. Kim is a theoretical and population ecologist who studies growth, spread, and extinction probability of small populations. So please join me in welcoming your panelists onto stage. Okay, so the question was basically, what are the implications and what do you think will happen if we eradicate all of these mosquitoes and these pathogens? Is it possible?
The short answer is no. We already tried with DDT and we failed miserably. But the eradication of a non-native species has somewhat different implications than the eradication of a species globally. So when we think about the Asian tiger, Aldopictus, or the species, they're not commonly found here. They're never found here, in fact. And so thinking about eradication of them in Canada, that seems like a reasonable, from an ecosystem point of view, a reasonable thing that you might do. Trying to eliminate something like Culex, that would have huge repercussions, negative probably on some ecosystems, and that's probably not something you want to do. So it depends on who you're talking about and where you're talking about when you're talking about eliminating a species. Does that help?
So the question is, why is it so particular to Brazil that we're seeing the microencephaly?
Everybody's looking at me.
Yeah.
Thank you. There actually were reports in the Micronesia and French Polynesia outbreaks of suspected uptickings of microcephaly and Guillain-Barré syndrome, but they were never teased out, and part of that was that it just wasn't big numbers. So they couldn't confirm that it actually was an increase, a significant increase, and they also didn't have good ways of testing that it was actually Zika virus that was in those particular cases. So there was a suggestion, it was noted that there was a possibility, but it really took Brazil when you got into a really big epidemic to really see clearly what the numbers were doing.
So the question was looking at groups outside of Brazil for the microcephaly.
I don't know what those numbers are for pregnancies in small places.
Yeah, outside of Brazil, I think the numbers are still fairly small. I think you're absolutely right there. So there is a question of what's going on in Brazil, and there are a lot of unanswered questions there. I mean, they're just identifying a case of microcephaly turns out to be a challenge, because what diagnostic criteria are we going to use? We know at the extreme end what they look like, but a lot of those 5,900 cases are diagnosed on the basis of just head circumference and what's the normal range there. There's all kinds of stuff there just about really just identifying what a case is that still has yet to be worked through.
The question for the audience was, are there long-term reproductive effects of being infected with Zika?
Yeah. Semen, sexual transmission, some reports have come out that there's a very high titer that builds up in semen. And what's not known is, even though the symptoms and signs of the disease disappear, nobody knows yet in reproductive tissues how long that virus might be hanging around. So we saw this kind of thing happen with Ebola, right? Thought it was another kind of an acute infection, and if you survived it, you cleared the virus. And that turned out not to be the case, where there was a reservoir in reproductive tissue, and it was semen happened to be the same thing. And I think in the ocular fluid they were finding resident Ebola. Virus was still viable and still live there, but these people were walking around and healthy. So we don't know yet.
So the question is, what's the estimation, what's the time frame, I guess, when this can move into a North American epidemic?
We're looking at you, Craig.
Okay, so
Well, I'll start, and Chris can help me out here. Well, it depends on a lot of things. First, you have to have a lot of infected people in the presence of the mosquito. You have to have a lot of mosquitoes biting those people, and so forth. So there are all those things that have to happen in order for it to really get continuing transmission. Chances are what you'll see in North America, if it in fact gets in Culex, and if in fact Culex can actually transmit the disease in the wild, it's only been documented in the laboratory under ideal conditions at this point. You're probably going to just see these stuttering epidemics. So somebody travels, to Cancun or whatever, and they, wear their flip-flops and get bit, and they get infected by an Aedes aegypti. They come back, and maybe you get a Culex who bites another individual, and so you might get a series of things happening. The ecology of the Culex, though, is of course quite different from Aedes. Aedes is a domestic mosquito. It loves to live around humans. Culex is more of a sylvatic or forest-dwelling species. So the contact between the humans and the Culex is not going to be as intensive as is the case with Aegypti. So there's a whole lot of variables there, and you can imagine how complex these when you start thinking of these different ecological factors. So the short answer is, who knows? Do you want to add to that?
I was going to say one of the bigger worries may be with climate change, and albopictus is coming really close to us. And we know that already harbors and can harbor not only Zika, but you saw a host of other flavi and some of the toga viruses too. So maybe we don't even need it to get into Culex to cause concern, because albopictus is coming our way.
So the question was, how effective is the GMO attack on these mosquitoes?
So there's already been some field trials with one particular kind of genetically modified mosquito. Just out of curiosity, how many of you have already heard of this Oxitec GMO mosquito? Okay, a few of you. Excuse me. I'm catching your thing.
Live demonstration, folks.
And they've done controlled releases in parts of Brazil. This is before Zika came around even, because they were using it to trial mosquito control because they were worried about dengue mostly. Dengue is a huge problem, much more so than Zika is. And so that's been released since 2013 in parts of Brazil and Panama, and is that it? Cayman Islands. And it was really successful, where they saw the populations of Aedes aegypti drop between 80 and 90%.
So the question was, is really GMO, is that the main attack, or are we researching all sort of three prongs of that attack that Mark was discussing in his presentation?
You have to do all three. You can't get away with just one branch. We just know that we can't completely get rid of the mosquitoes forever. So, I mean, you know, if you say 90%, you still have 10% there, right? Great reduction. You're minimizing the risk, but it's not down to zero.
So I'd just add to that, you know, we almost got rid of Aedes aegypti from the New World in the 1940s. We had a major campaign, and of course part of that was DDT, but that wasn't the only thing that was done. So that we were spraying oil on water. There was very aggressive campaigns around, you know, covering water sources and that sort of thing. So we were very successful in the 1940s, and of course, like many public health campaigns, we're successful, so why do them anymore? And of course, then we had problems with pesticides, and the mosquitoes did become resistant to DDT. But it came roaring back, mostly because we stopped doing what we were doing, which was really sort of low-tech, integrated pest management strategies. I mean, you put screens on people's windows, you cover water supplies. You know, the Aedes aegypti is a perfect mosquito for a throwaway society because it lives in trash, it loves trash. So, you know, it's pretty basic environmental stuff that can be done to control these mosquitoes. And one piece of it is the GMO, and that certainly contributes to it. But we have lessons from the past that these sort of low-tech kind of integrated strategies, in fact, are quite successful. Work with malaria mosquitoes, it worked with Aedes aegypti as well.
So what percentage of the general population in Brazil has been infected by Zika?
Nobody knows. Yeah.
I don't know those numbers.
There's no way really to know that, because, you know, if you have 80% non-symptomatic, you know, it's anybody's guess, really.
A lot.
Yeah.
So the question really is there a process for screening of those coming back to see, you know, are they infected with Zika, and what we can do about that?
So the current test for Zika is involving, it's called reverse transcriptase PCR, where they can actually amplify the viral genome, but you're only going to be able to do that when people are actively infected with the virus. And you saw there's a really short window. The other way to do it is to look for antibody that they make to the virus. And this has been complicated in the past because you saw that Zika virus is closely related to dengue and yellow fever and some other viruses. So people who are living in certain areas of the world, tropical and subtropical areas, often have antibodies to some or all of these things. So tests for very specific antibody that's unique to Zika virus, those tests are just being worked out. And one thing that kind of narrows the field as to, well, where is this person in their infectious cycle if they can't find live virus, is to look for a particular class of antibody called called IgM. So it's the first kind of antibody that you make. And once you make that antibody, it only hangs around, and for Zika, 30 days. So if they screen people and find there's evidence of Zika IgM, they know that they've been exposed to the virus and have had an infection, be it symptomatic or asymptomatic, within the last 30 days. So that kind of helps to see where they were and who these people are.
It's a historical view, but it doesn't speak to reproductive.
So the question really is, how long does it hang around? Really, I think it's more, if you have an immunity to this, does it persist and can it have effects afterwards?
Well, no, they do know that in people who get the course of disease that I showed you there, it's short-lived. I think the longest that they could actually show that anybody was showing symptoms and carrying the virus was about 12 days. No, in the blood. The semen thing is very new, and so who knows? No one knows yet quite how long it can still hang around in semen. And so that's still, that's an area of caution, certainly. But, you can tell if people have been exposed, infected with the virus. And so if you're thinking of getting pregnant with this person, beware.
So the question is, if a person likely would be infected with the virus shows no symptoms, can they still pass that on in through sexual transmission?
I don't know what the levels of Zika virus, I don't know that it's even been reported, the levels of Zika virus in people who are asymptomatic, because they don't come on the radar of the researchers who look at that kind of clinical test yet. But the IgM antibody test would tell you who those people are who were asymptomatic, yet they were exposed to the virus, and you could start to dig into them possibly and start looking for evidence of the virus still in the semen. So that's really in its infancy still. Lots of questions without answers.
Do we have any anecdotal evidence, like the one sexual transmission case that was very early? Did they continue to monitor how long that virus hung around?
Yeah, that's the Texas
Well,
case.
not the Texas one, but from the initial case, they came back and they were out for about 8 days, back home for 8 days, being asymptomatic, before actually showing signs. So assuming that the mosquito didn't travel with them, that means that there was 8 days since they were bitten before they actually started even being symptomatic. And in that period, that's when they actually had intercourse and gave it to their partner. It's not exactly the same because they actually eventually did develop symptoms. So those people that remain asymptomatic, I think what you bring up is truly the biggest problem right now, because that's the unknown. And getting information on the unknown, because you don't know where to look for it, it's going to be very hard unless we test everybody that goes and travels to these regions, for example.
Is it a male to female infection, or can it go the other way from female to male?
Let's put it, it's from male so far.
Yeah.
Yes. So the question is it suspected that the neural defects occur in the first trimester? Is that the sort of the critical window of when these things occur?
And I think that information is out there. I didn't really look carefully at it. Yeah, that's a really good question, and something that is probably Googleable.
Mm-hmm. I think that is the case, though.
Was the dengue vaccine commercially available globally or anywhere else?
So far, it's limited to Mexico right now. And eventually it'll get approved in other countries, but it's not yet.
Or it's just waiting on
Approval.
because there's a dengue outbreak in Mexico, which is probably why they needed it, wanted it to try.
So the question was comparing something like Zika to Ebola. How do you think the stealthiness of Zika will limit the abilities to stop the spread of the disease?
So the question, just so that we're clear on it, is can Zika become more pathogenic?
So we're looking at priorities, I guess. Is Zika a higher priority than something like Ebola or maybe, other related mosquito-borne diseases?
I mean, well, WHO was much quicker to jump on the Zika case than it was on Ebola. And probably the reason it did declare a public health emergency so quickly was because it took so long to get its act together around Ebola. That's not really answering your question. But WHO certainly brought this, made it a high-profile situation. And I think the issue around Guillain-Barré, as well as particularly microcephaly, and the consequences, the long-term consequences that this has on families and so forth, that those are, even though it's rare, it's still very profound. So I think that it probably will stay on the radar for that reason. And I think there, as many of the questions here have certainly indicated, there's a whole lot of stuff we really don't know yet about Zika. I think it depends a little bit on what the research begins to tell us about things, whether it can become a widespread person-to-person transmission sexually or not. There are some of those things that may actually keep it very much in the public eye.
The thing that's interesting about Zika is that you really can group it with dengue, with yellow fever. And so if you're combating one, you're essentially combating a lot of them, because a lot of the same approaches are going to be applicable to all of those. And so, in a way, even if we lose sight of Zika, we might not lose sight of dengue, yellow fever, and so we're going to be combating it in one way or another. Whereas Ebola is really unique.
The question was, do we know with babies infected or that have the microencephaly, do we know, are they cleared of the virus, and what is their outcome predicted to be?
I don't know, that information may be out there. I haven't looked. There's a fairly large proportion of fetuses that die in utero. And die shortly after birth too, because the deformity and the impacts of it can be really profound. Some of the microcephaly cases that I've been reading about too went unmissed because there are a proportion of young fetuses where the part of the brain that isn't developing and pushing that skull up fills with fluid, and so you do get a normal or a near normal head circumference, and so some of those cases were missed as well, and they only saw it because they looked on ultrasound and saw that the brain was half the size it should be. So there's still a lot of diagnostics that need to be done and how they're going to detect all these different cases. So I can't answer all of your question.
Do you think we'll see more social reforms in relation to the high incidences of microencephaly that we've seen in Brazil?
That's a really good question. I mean, it'll certainly provoke a lot of, discussion. And whether these translate into sort of policy reforms or policy changes, I think will wait to be seen. I mean, there's some, was it El Salvador, I guess it was, that say, well, women should just not get pregnant for 2 years. Just, let it become endemic, well, you'll be immune, and then you can start again. I mean, that so far has been about as much response as we've seen, but it'll be interesting to see how this discussion goes forward, because it really will start to revolve around reproductive rights and so forth. And I think that this is a case where you might have one of the earlier questions, this might actually be where the disease propels perhaps a more difficult social discussion or social response that might have impacts way beyond the disease. It would be interesting to keep our eye on that.